Hi, I'm Brianna, and I'm doing acetylcholinesterase for my biology protein model project. This protein basically functions enzymatically by catalyzing hydrolysis of acetylcholine and into choline and acetate at a cholinergic synapse. This activity is vital for regulating neurotransmission in the nervous system, and it also plays a crucial role in facilitating biochemicals reactions necessary for proper function of the nervous system. This protein is usually widely distributed enzyme crucial for neurotransmission and neuromuscular functions across the various organisms. So it's typically located in the synaptic clefts, particularly at the cholinergic synapse. It's closely associated with the postsynaptic membrane as well. These synapses are prevalent throughout the nervous system, notable at neuromuscular junctions and within the central nervous system. Neurons, including motor neurons and those involved with cognitive responses, commonly contain this protein to regulate the acetylcholine levels as well. A neurotransmitter essentially for the muscle contractions, sensory perception, and cognitive function. Its presence extends beyond mammals as well, into birds and reptiles and amphibians, insects. So as you can see, this is my model of this protein. The red represents the beta plated sheets, and then the blue represents the alpha helixes, and the white parts are just extra holding everything together. And this right here is my active site. Usually this is in the middle. This is in the middle of the model, but for this I'm just gonna keep it like this so it's easier to explain. Alright, so I put it on a piece of paper, and this blue site right here is the anionic site where hydrophilic molecules bond, and this green part right here is where the hydrolysis occurs and the molecules are broken apart. And so I got my acetylcholine right here. This red part will bond to this anionic bonding site, and this blue part right here will bond here, and basically it'll be hydrolyzed by a water molecule, and the bond will be broken down. And after the acetylcholine is broken down, it will turn into a choline molecule and also acetic acid molecule. So basically what I talked about is if the protein were to be functioning properly. However, there are times where it can have mutations. The mutations involved the binding of serotonin toxins to the active site of this protein, which would make the protein less active. The serotonin toxins inhibit the enzyme activity by binding inversely to its active site, thereby preventing it to hydrolyze the acetylcholine. As a result, this can mess with the ability of the catalyst to break down the acetylcholine and cause it to be impaired, leading to accumulation of acetylcholine at the cholinergic synapses. This accumulation can overstimulate the cholinergic receptor and distribute normal neurotransmission, ultimately resulting in symptoms such as muscle paralysis and respiratory failure. Therefore, the mutation caused by the serotonin toxins bonding reduces the activity. So this is our active site right here, and this part right here is where the serin is going to bond, which causes a problem because if you have the acetylcholine trying to bond here, it will only be able to bond to this anionic site, which is this blue part right here. However, it will not be able to bond to this site right here, which is where the hydrolysis occurs, which leads to problems because it will not be able to break this down. However, there can be like a little bit of breaking down for the anionic site. There's not going to be much breaking down of the other site where it's hydrolyzed. So this is going to cause a lot of problems with communication and for the body, which can ultimately lead to really damaging effects. The main functional group present in the original amino acid side chain is the hydroxyl group of serine. Serine's side chain is hydrophilic and neutral, has a neutral pH, contributing to its ability to form hydrogen bonds with water molecules and participate in protein folding and stabilization. Additionally, serine, relatively small chain, enables it to fit into protein structures without causing steric hydrants. In contrast to the mutated amino side chain resulting from serine toxin binding is the phosphorylate group, which is hydrophilic and it's negatively charged and larger compared to the serine, influencing its interactions within the protein structure. Specifically, the group's larger size and negative charge can lead to altered steric interactions and electrostatic interactions, potentially disturbing the protein stability and function. The mutation causes effects in the protein's function. As serine toxins inhibit the enzymatic activity, it leads to acetylcholine accumulation at the synapses, which disturbs the neurotransmission and can cause severe physiological consequences at the cellular and organism levels, including respiratory failure and even potential death.